Hello friends, welcome to my channel Botanical Studies. In this video, we will discuss about lipids. The discussion will go through following points: introduction of lipids, structure of lipids, classification of lipids, properties of lipids, and finally significance of lipids. Introduction of lipids. Lipids are a heterogeneous group of organic compounds that are insoluble in water and soluble in non-polar organic solvents. In the human body, lipids can be synthesized in the liver. Lipids are also found in oil, butter, whole milk, cheese, fried foods, and also in some red meats. They are naturally occur in most plants, animals, and in microorganisms. Lipids are used as a cell membrane component, wax coating material that protects plants, energy storage molecule, insulation against cold, and it is also act as a hormone. Structure of lipids. Lipids are made up of elements like carbon, hydrogen, and oxygen, but have much lower properties of water than other molecules, such as carbohydrates. Unlike polysaccharides and proteins, lipids are not polymers. They lack a repeating monomeric units. They are made from two molecules, glycerol and fatty acids. A glycerol molecule is made up of three carbon atoms with a hydroxyl group attached to it and hydrogen atoms occupying the remaining position. Fatty acids consist of an acid group at one end of the molecule and a hydrocarbon chain, which is usually denoted by the letter R. The fatty acid may be saturated or unsaturated. A fatty acid is saturated if every possible bond is made with a hydrogen atom such that there exist no carbon double bonds. Unsaturated fatty acid, on the other hand, do contain carbon-carbon double bonds. Monosaturated fatty acids have one double bond in between carbons and polysaturated have more than one carbon-carbon double bond. Let us discuss the difference between saturated fatty acid and unsaturated fatty acid. Saturated fatty acids, they are commonly called as fats, while unsaturated fatty acids, they are commonly called as oils. Saturated fatty acids contains a single bond in between the carbon atoms, while the unsaturated fatty acid contains at least one double bond in between carbon atoms. Saturated fatty acid not to be consumed more than 10% of total calories per day, while unsaturated fatty acid not to be consumed more than 3% of total calories per day. Excessive consumption of saturated fatty acids leads to heart diseases, while unsaturated fatty acids are good for consumption, but excessive may increases cholesterol level. Saturated fatty acid increases low density lipoproteins, which is called as bad cholesterol, while unsaturated fatty acid increases high density lipoproteins, which is commonly known as good cholesterol, and also reduces low density lipoproteins. Saturated fatty acid would not spoil quickly, but unsaturated fatty acids spoils quickly. Food sources of saturated fatty acids are whole milk, butter, cheese, margarine, coconut oil, vegetable oil, meat, peanut, fried foods, etc. While the food sources of unsaturated fatty acids are walnut, flax, avocado, sunflower oil, soybean oil, fish oil, canola oil, red meat, etc. Saturated fatty acid have high melting point, while unsaturated fatty acid have comparatively low melting point. Saturated fatty acid is found in solid state at room temperature, while unsaturated fatty acid is found in liquid state at room temperature. Myristic acid, palmitic acid, and stearic acid are the examples of saturated fatty acid, while oleic acid, linoleic acid, and linolenic acids are the examples of unsaturated fatty acids. Myristic acid having chemical formula CH3CH212COOH. Palmitic acid having chemical formula CH3CH214COOH, while stearic acid having chemical formula CH3CH216COOH. Unsaturated fatty acid that is oleic acid having chemical formula CH3 CH2 7 CH double bond CH CH2 7 COOH. Linoleic acid having chemical formula CH3 CH2 4 CH double bond CH CH2 CH double bond CH CH2 7 COOH. Then linolenic acid having chemical formula CH3 CH2 CH double bond CH CH2 CH double bond CH CH2 CH double bond CH CH2 7 COOH. That is oleic acid with single double bond in between carbon atom. In case of linoleic acid, there is two double bond in between carbon atoms, while in case of linolenic acid, there are three double bonds in between carbon atoms. Classification of lipids. Lipids can be classified according to their hydrolysis products and according to similarities in their molecular structures. Lipids are classified into four different ways: simple lipids, compound lipids, steroids, and prostaglandins. Simple lipids are further classified into fats and oil and waxes, while compound lipids are classified into phospholipids and glycolipids. Phospholipids are glycerophospholipids and sphingophospholipids, and glycolipids are classified into cerebrosides and gangliosides. Steroids are further classified into cholesterol, bile salts, steroid hormones, that is testosterone and estrogen. Simple lipids. They contain two types of compounds, a fatty acid and an alcohol. Fats and oils, which yields fatty acid and glycerol upon hydrolysis. Both types of compounds are called triglycerol because they are the esters composed of three fatty acids joined to glycerol, that is trihydroxyalcohol. Fats. Fats are triglycerides that are solid at room temperature, usually derived from animals, contains mostly saturated fatty acids. Oils are triglycerides that are liquid at room temperature, usually derived from plants or fish, contains mostly unsaturated fatty acids. Waxes. Wax is an ester of long chain alcohol and a fatty acid. On hydrolysis, waxes yield fatty acid and long chain alcohols. Waxes are insoluble in water and not as easily hydrolyzed as fats and oil. They often occur in nature as a protective coating on feathers, fur, skin, leaves, and fruits. Waxes are used commercially to make cosmetics, candles, ointments, and protective polishes. Sebum, secreted by sebaceous glands of the skin, contains wax that helps to skin soft and prevent dehydration. Second group of lipid is a compound lipids. Phospholipids, which yields fatty acid, glycerol, phosphoric acid on hydrolysis. They may be glycerophospholipids or sphingophospholipids, depending upon the alcohol group present in it. First, glycerophospholipids. They contain glycerol backbone, two fatty acids, and phosphoric acid group. For example, lecithins. Lecithins are widely distributed in cells and are most abundant phospholipids. It forms an important structural component of cells, membranes, form cells, which play a role in the transport of lipids in the bloodstream. Commercially, lecithin extracted from soybean is used as an emulsifying agent in margarine and candies to provide smooth texture. Sphingophospholipid. Sphingophospholipid are complex lipids that contain sphingosine instead of glycerol. One important type of sphingospholipids are the sphingomyelins. Sphingomyelin on hydrolysis yields sphingosine, a fatty acid, phosphoric acid, and choline. Sphingomyelin are found in brain and nerve tissue and in the myelin sheath that protects nerves. Second compound lipid is a glycolipid. Glycolipid includes two major groups, cerebrosides and gangliosides. Cerebrosides, they are abundant in myelin sheaths of nerves and white matter of the brain. They are based on spingonin and contain In addition, a fatty acid like behenic acid, lignoceric acid, nervonic acid, cerebronic acid, and a monosaccharide sugar galactose without phosphoric acid or glycerol. Gangliosides. They are based on ceramides. The ceramides are linked to carbohydrates and also contains a non-acetyl nuraminic acid. Gangliosides act as antigens. Third group of lipid is the steroids. Steroids are classified as lipids because they are soluble in non-polar solvents, but they are non-saponifiable because the compounds are do not held together by ester linkages. The basic steroid structures contains four fused rings. There are three different types of steroids: cholesterol, bile salts, and steroid hormones. Cholesterol is the most abundant steroid in the body. It is an essential component of the cell membrane and a precursor for other steroids such as bile salts, sex hormone, vitamin D, and adrenocorticoid hormones. There is apparently a correlation between high levels of cholesterol in the blood and atherosclerosis. Bile salts. Bile is a yellowish brown or green fluid produced in the liver and stored in the gallbladder. Bile salt act like soap and other emulsifiers. They contain both polar and non-polar regions, helping to break fats in foods into similar pieces, allowing them to hydrolyze more easily. Steroid hormones. These are the chemicals released by cells or glands in one part of the body that send out messages that affect cells in other part of the body. Many hormones are based on steroids. Sex hormones like testosterone and estrogen produced in the testes and ovaries regulate the production of sperms and eggs and aid in the development of secondary sex characteristics. Fourth and last group of lipid is the prostaglandins. Prostaglandins are derivatives of fatty acids. They are synthesized from arachidonic acid. They were first discovered in seminal fluid, hence the name prostaglandins. They are also recorded from tissues such as kidneys, testes, placenta, uterus, stomach, lungs, brains, and heart. Like hormones, they are involved in body processes including reproduction, blood clotting, inflammation, and fever. Let us discuss the physical properties of lipids. Lipids may be either liquid or non-crystalline solids at room temperature. Pure fats and oils are colorless, odorless, and tasteless. They are energy-rich organic molecules. They are insoluble in water but soluble in organic solvents like alcohol, chloroform, acetone, benzene, etc. They have no ionic charges. Solid triglycerol, that is fats, they have high proportion of saturated fatty acids. Liquid triglycerol, that is oils, they have high proportion of unsaturated fatty acids. Chemical properties of lipids. Hydrolysis of triglycerol. Triglycerol, like any other esters, react with water to form their carboxylic acid and alcohol, a process known as hydrolysis. Saponification. Triacylglycerol may be hydrolyzed by several procedures, the most common of which utilizes alkalis or enzyme called lipases. Alkaline hydrolysis is termed saponification because one of the product of the hydrolysis is a soap, which is generally sodium or potassium salts of fatty acids. Hydrogenation. The carbon-carbon double bond in unsaturated fatty acids can be hydrogenated by reacting with hydrogen to produce saturated fatty acids. Halogenation. Unsaturated fatty acid, whether they are free or combined as esters in fats and oil, react with halogens by addition at double bonds. The reaction results in the decolorization of the halogen solution. Rancidity. The term rancid is applied to any fat or oil that develops a disagreeable odor. Hydrolysis and oxidation reactions are responsible for causing rancidity. Oxidative rancidity occurs in triacylglycerols containing unsaturated fatty acids. Finally, let us discuss about the significance of lipids. It is established that lipids play extremely important role in the normal functions of a cell. Not only do lipids serve as a highly reduced storage form of energy, but they are also play an intimate role in the structure of cell membrane and organelle membranes. Lipids perform many functions such as energy storage, making biological membranes, insulation, protection, for example, protecting plant leaves from drying up, buoyancy. It also acts as a hormones. Lipids are also act as a structural component of the body and provide the hydrophobic barrier that permits partitioning of the aqueous contents of the cell and subcellular structures. Lipids are major sources of energy in animals and high lipid containing seeds. Activators of enzymes, for example, glucose 6 phosphatase, steryl coenzyme A desaturase, and omega monooxygenase and beta hydroxybutyric dehydrogenase requires phosphatidylcholine micelles for activation. So this is all about